Hi and welcome to this video. We're going to be running through all of the OCR module 4.2, which is on biodiversity. Now this module, if you're watching this in 2022, can be assessed in the upcoming exam. So it's going to be really important to understand this. I'm going to be breaking it down into these major areas. So for today's mind map, a little bit different. We aren't actually going to need any resources. We're just going to need a big piece of A3 white paper. And you can start by kind of laying out these headings in this kind of orientation. So we're going to talk about the levels of biodiversity, how to sample for biodiversity, and how we can calculate and measure biodiversity. Then we're going to talk about what are the benefits of biodiversity, what are we doing that affects biodiversity, and ways we can conserve biodiversity together with laws around biodiversity. And I'll give you a little sneak preview because I've already done this mind map once. It's going to look a little bit like this, and then it's going to look a little bit like this. Okay, let's get started. The first thing we're going to look at is the different levels at which we can assess biodiversity, and I'm going to zoom right in so I can write clearly for you. So we can look at the levels of biodiversity in three separate ways. We can look at measures of genetic biodiversity. We can look at habitat biodiversity. And we can look at species level biodiversity, which is probably the most important one to consider. But first we'll look at genetic. So genetic biodiversity is really just looking at how much variation there is in alleles within a species. So we can define it as this. It's the variation between individuals belonging to the same species and we're talking about really alleles. So when we measure genetic biodiversity, what we look at is the percentage of polymorphic gene loci. So let's say an organism has 20,000 genes. So for example, if 1,000 out of 20,000 genes have at least two or more alleles, then we could work out the percentage genetic biodiversity of that species. So this calculation would be 1,000 divided by 20,000, I'll just simplify to 1 divided by 20, which would equal 5%. So this organism, which out of its 20,000 genes, only 1,000 of them have 2 or 3 or 4 alleles, it has a measure of sort of 5% genetic biodiversity. So that's one way of looking at biodiversity. The second thing is looking at habitats. So we can look at the habitats in an area and kind of say, well, how many different types of habitat are there? So it is the range of different habitats within an ecosystem. So I always like to think about this one in terms of like going to your local park. So in my local park, there's an area with a lot of trees. It's kind of like a mini woodland. That's one kind of habitat. And there's 2 ponds, in fact. So that's kind of like an aquatic habitat. And then there maybe is an open field area, which is more of a grassland habitat. So it's the range of different kind of mini habitats within a larger environment. And the final way we can consider biodiversity is species biodiversity. We can actually break this down. We can break this down into 2 things. We can break this down, I'm going to use a different color, into species richness and evenness. So richness and evenness. So species richness is the number of species in an ecosystem. Whereas species evenness is how evenly represented the species are in that ecosystem. So, for example, if you went to the Amazon rainforest, it's a very biodiverse place. It has high richness and high evenness. There are a lot of different species, and actually, if you looked at like 100 meters square of rainforest in the Amazon, you might not even find the same species of tree twice. Every single tree will be a slightly different species, and they'd all sort of be evenly spread, jostling for position. So that would be a very rich and even kind of habitat. One habitat might be very rich in species. There might be 30 different species there. But if you had just one type of species way in abundance and all the other species much lower numbers, then it would be rich but not even. All right, so when we're looking at biodiversity, we need to actually calculate biodiversity. If we're trying to assess the richness and the evenness of an ecosystem, in terms of the species biodiversity, we have to do some sampling, and with that sampling we can do some measuring. So I think I'm going to talk about how we can look at richness and evenness by measuring, and I'm going to go up here. So I'm going to zoom up to this area and talk about measuring, and I'm going to slide across, and we're going to talk about Simpson's index of biodiversity. So Simpson's index of biodiversity takes into account both the species richness and evenness. So it's a measure of species level biodiversity. Now hopefully you're coming to this video as a bit of revision, so you know a bit about it already, but we might calculate it using a table. So, for example, if we had a table here which had the species, and over here we had percentage cover of the sort of average percentage cover of some quadrats. And maybe we're looking at some different species in our quadrats. We're looking at grass, we're looking at clover, and we're looking at dandelions. I think I spelled that wrong, dandelions. So let's say that in field A and field B, there are different percentage covers of these species. So what values are we recording? Well, let's say that we've got 98% cover of grass in field A, 2% cover with clover, 0% cover with dandelions. So probably already you can see that field A is not very evenly, the species aren't evenly weighted. There's an abundance of grass and very little of anything else. Now let's say field B has got 33% of each, like this. So there's 3 species and they're roughly evenly weighted, 33 and a third, let's say. So the total percentage cover in each place is 100. They add up to 100, so this is the total. Now, how would we measure the Simpson's Biodiversity Index? Well, we need to know the formula. Well, actually, you get given the formula, but you have to be able to use it. So D, which is the value for Simpson's Biodiversity Index, is equal to 1 minus, and then you've got a big bracket here, and it is the sum of small n divided by big N squared, like that. So what this means in terms of percentage cover is it's 1 minus the sum of basically the percentage covers squared. So n for percentage cover is just, let's say, 98. It's the percentage cover of one species. And big N is the total percentage cover, which is going to be 100 in this case. If you're looking at animals, so let's actually just label this here as well, because I think this is important. So N equals, it's the number of animals or percent cover, and big N equals the total number of animals, all of the different animals, of all the different species, or if it's percentage cover, it's generally 100, okay? So in this instance, if we were working out the biodiversity index of field A, let's actually do this. So A equals 1 minus big brackets and the sum of all these things. So I'm going to do each step. So it would be 98 over 100 squared plus 2 over 100 squared plus the last one is 0 over 100 squared, so that's just 0, okay? So it's 1 minus 98 over 100 squared plus 2 over 100 squared. Now that's going to be a number, I'm going to get on my calculator, so it's 0.98 squared, so let me just do that, 0.98 times 0.98 equals, so it's 1 minus 0.96, that's the whole thing, plus the other one is 2 over 100 squared, so it's 0.02 times 0.02 squared, that's like plus 0.0004, 0.004, so I add that to my 0.96, which is 0.9604, but it's 1 minus that, so 1 minus 0.9604 equals a really, really small number, 0.0396. That is a low value for field A. For field A, I've got a low value of Simpson's biodiversity index. For field B, it would be much higher. If you want to work it out, please be my guest, but I'm not going to. But let's just say what figures we can get, what range of values we can get. So 1 is the highest. The highest you can ever get is 1. And it's something like the Amazon rainforest would have a Simpsons biodiversity index of approaching 1, probably wouldn't even be 1, or like a coral reef somewhere would be, you know, something like that. And 0, I've got all line through so you can see, 0 is the lowest. Now this field here was pretty close to 0, but something with 0 would be like a monoculture crop field. So monoculture means like all a huge field with just corn. And if you think about the maths of it, that would be 0, because the only thing you'd be measuring is corn. It would be 100% percentage cover. So we're doing 1 minus 100 over 100 squared. What's 100 over 100? It's 1. What's 1 squared? It's 1. What's 1 minus 1? It's 0. Okay, so if you had a monoculture, you'd have a Simpsons biodiversity index of 0. Okay, so that's Simpsons biodiversity index done. So we talked a little bit about percentage cover and how we sort of, well, we didn't really talk about how we get this percentage cover data. Well, we'd obviously have to do some sampling. So we're going to talk about sampling now. So we're going to move across, and here we go. So when we talk about sampling, we can either talk about sampling animals, or we can talk about sampling plants. Okay, and these are different textbook pages of your textbook, most likely. So first of all, let's talk about sampling plants first of all, because we've been talking about percentage cover, so that makes sense. Now, when we're looking at plants, often we're going to be using quadrats. Often. So here's a picture of a quadrat in use, just in case you're not sure what it looks like, but there's a quadrat. And when we're using quadrats, there's basically two ways we can use it. We can either basically do some counting and just count the number of plants that is in a quadrat, or we can do percentage cover. Now this quadrat here has lots of little sort of sub-squares which help to estimate the percentage cover. And often each sub-square in a quadrat, depending on the exact quadrat, might be 1/100th of the total area. So it helps you kind of estimate the percentage cover in terms of how much of the quadrat is grass and how much of the quadrat is clover, etc., etc., etc. So when we're using quadrats, the first thing we might have to decide is whether to use them in a random or a non-random way. So, random and non-random. So if we want to randomly use quadrats, basically what that means is we've got a big old area that's kind of roughly the same, and we want to just get a feel for what is out in that area. So you've got a field that is relatively homogeneous, which means it's basically all one kind of the same kind of area. And we just want to kind of go out there and throw some quadrats around and see what's out there. We don't just go out and throw them around because that might be biased. So we want to use some sort of other better method. So I would write, first of all, we could use I'm going to go a bit more zoomed in. Could use random throwing. But there's a possibility of bias. You might not be aware of the bias, kind of unconscious bias, I suppose. You might think you're throwing them randomly, but actually secretly you're kind of throwing them all in the direction of an interesting plant that you want to go and look at. So it's much better to use a random number generator. And a random number generator, you will probably have one. Basically, well, if you have a calculator, if you have a phone calculator like this, if you turn it sideways, there's a button there that says rand. Where is it? There it is. And if you press it, it gives you a random decimal between 1 and 0. And what you can do with that is you can use it to give you a coordinate. So you divide up your field, let's say, from 0 to 1 on this axis and 0 to 1 on this axis. You press rand, it takes you to a point somewhere between 0 and 1 on this axis. You press rand again, it takes you to a point somewhere between 0 and 1 on this axis. You go to where the 2 points meet in your grid, and you go and sample that specific coordinate. So a random number generator can generate coordinates. And the main positive of this is it can avoid bias. Now the negative of this whole random method is it may underestimate your biodiversity if your habitat is not homogeneous. So if actually your habitat is not just one uniform field, but it's a field with a kind of sandy, kind of boring area, and maybe a more marshy, more interesting area, then it might not be appropriate to use random sampling. So the negative is it may underestimate. If you miss out part of a habitat. So if the habitat is not uniform, then you've got to use some form of non-random sampling. So I'm going to slide over here and we're going to focus on that. So non-random sampling can be opportunistic, it can be stratified, or it can be systematic. So let's look at each one in turn. First of all, opportunistic. So opportunistic sampling is when you can kind of easily see, or you have prior knowledge about a habitat, and you know it can be kind of divided up into sub-areas. So it's based on prior knowledge. Change color. And it's pretty quick and easy. But it may be biased. So this opportunistic sampling is maybe you know that your park's got a, it's got a pond and it's got a field, and you want to quickly do some sampling in the field. So you go into the field and you throw some quadrats around, and in the pond you want to sample the pond life there. Maybe it's a shallow pond. You throw some quadrats around, you see what aquatic plants are there. So maybe you decide to do like 10 in the field and 10 in the pond. But it's a bit, it's quick and easy. It's a bit, it's not very rigorous. It's a bit slapdash. So we can kind of make it a little bit better than just being opportunistic. And there's a kind of next level, which is a little bit better. And that's called stratified sampling. So what is stratified sampling? Well, this is when you divide up the area into zones that appear distinct. And then what you do is you sample all the areas. And typically you should do this according to the ratio of areas. So maybe you've got a field and you know that 40% of the field is very dry, 40% of the field is a little bit hilly, and 20% of the field is marshy. So if you know it's 40, 40, 20, you would also want to split up your quadrats. Let's say you did 100 quadrats, you'd also want to split them up in a 40, 40, 20 ratio. So we should write that down. Divide up the area into zones that appear distinct. All areas sampled. And we could put according to the ratio of areas. So there was a recent exam question on this with the paper that I sent my students, and it had like a big old map that was like this or something, and it basically had, on the map, it basically said that there were like 3 areas. It was like A, I don't know, well, A, B, and C, but it gave you the meter squared areas of A and B and C, and it had them clearly labeled as like, let's say A was a forest, and I can't remember what C was, grassland, and B was whatever. But then it asked you, what type of sampling would you use? Answer, stratified. And how would you do it? And you had to do some maths to calculate the ratio of areas and say how you would work out the number of quadrats to do. All right, the final type of non-random sampling is systematic sampling. And systematic sampling takes samples at fixed intervals. So that's kind of the systematic part. You basically move along systematically, sampling and sampling and sampling. And typically you do this along a transect. Now transects can either be line transects or belt transects. And they both do roughly the same thing, which is to measure changes in distribution along an axis. So what do I mean by this? Let's say, I'll just draw another little picture here. Let's say that you've got a little pond. Here it is. And the pond kind of is a bit marshy. It has marshy borders, okay? So near the pond is quite sort of marshy ground, plants growing. And then as you move away from the pond, it gets drier. And then perhaps away from the pond, we start getting more kind of typical foresty stuff. Excuse my drawing here. And you might want to see how the plants change moving away from the pond. So you might lay a line down on the ground like this. That's your transect line. And you might take a quadrat first here, move down, then here, then here, then here, then here, and so on. So you get your percentage cover for different plants at 0 meters, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And you see, do the plants change as you move away from the pond and the ground dries out? Answer probably yes. Okay? So, let's pinch out and see what we've covered so far. So far, we've looked at the levels of biodiversity, Simpson's index of biodiversity, and sampling plants. But we almost forgot, or I almost forgot, to look at sampling animals. So let's go there next. So, animals, we don't tend to use quadrats. Reason? Animals don't really stay still. So if you throw a quadrat at an animal, it will run away, and then you won't be able to identify it. So we don't really use quadrats with animals. There's a couple techniques that you need to be able to talk about. So the three that I'm going to focus on are, first of all, pitfall trap. Second of all, sweep net. And third of all, something called a Tolgren funnel. Okay, so let's just zoom in a little bit on these. First of all, pitfall trap. Here's a diagram of a pitfall trap, and we can see that it is a place where an insect can walk and drop into a little pot or a little container. Now you put this pitfall trap in the ground, you dig a little hole for it, and you make sure that the lid of it is basically level with the surface of the ground. Unsuspecting insects can wander along and in they drop into the little cup and they can't get out. You might potentially lay leaves over the surface of the pitfall trap so the insects can't kind of see it coming. And then you leave it for a few days and you come back and you sort of have a look what insects you've collected. Another technique is a sweep net. Here's a picture of someone sweep netting. Now a sweep net is basically a long handle and it's got a net on it, and you basically move it backwards and forwards. You could move it across long grass and you try and sweep up the bugs off the leaves of the grass. You could also do it in a pond or through the branches of a tree. And then you do some sweeping backwards and forwards, and then you look in your bag and you can see that there are some insects there, and then maybe you can tap them out into a tray to try and identify them. Finally, there's a Tolgren funnel. This is slightly different because this one actually kills the insects. You basically put it under a tree, you put a bunch of Tolgren funnels under a tree, and the funnel releases insecticides, kind of gas, that goes up into the tree, kills the insects in the tree, but they fall down and land in the funnel. Or there's another version of this as well where it's kind of like a lamp, and the insects are attracted to the lamp and then they die and they land in the funnel. So the difference between these, the main difference is the Tolgren funnel is, kills the insects. Whereas the other two methods basically give you live insects, well, potentially, if you go and get them soon enough. If you just leave them in the pitfall trap, they might die. So with these two methods, you can have live insects, and that means you can do catch and release. Now, catch and release uses live insects or live of any organism to try and estimate the size of the population. Now, there's some great videos on catch and release. A particularly good one, if you Google it, catch and release ants, shows how you can catch a little ant, paint it with a fluorescent dye, and then release it, and then catch it again. So if you watch that video, you'll understand how to do it. But basically what you do is you catch a certain number of animals, you mark them in some way that you can later detect, and then you let them out. And then you let them wander about, and you come back maybe a day or so later, once they've moved around and mixed in with all their other kind of buddies in the population. And then you do another catch, and you look at how many of the second catch has the mark that you put on them first time round. And then you can do some maths. So the maths is this. Let's just put a different color. Let's do blue. So C1 times C2 over C3. Actually, I'm going to call it CM. CM. Sometimes it's written as C3, but I'm going to call it CM. So what do these things mean? Well, the top one, C1, is, let's keep it, let's make it green. So top one, C1, is first catch size. So maybe the first time round, you caught and you marked, I don't know, 50 ants, let's say. 50 ants you painted with a little dye. C2 is the second catch size. So maybe the second time you went back, a couple of days later, you caught 50 ants again. So in which case C1 and C2 would both be 50. Or if you caught 50 the first time, and then the next time you couldn't find as many and you only managed to catch 40, then you do 50 times 40 above. And what's underneath? Underneath CM is the number of the second catch who are marked. So that will give you an estimate of the population size. I'm not going to go too deep into the maths, but it's basically like this. If you label loads of ants first time round, you come back and only one of the second catch is marked, well, that must mean that the population is huge. Let's say you labeled 50 ants first time round, but actually when you caught them again, only one of those marked ones turns back up. That probably means that you only labeled a very, very small percentage of the population, so that, i.e. the population is huge. However, if first time round you labeled 50 ants, and the second time you come back and you caught the same number, but this time, half of them were marked, 25 of the second catch were marked, well, that probably means that the first catch, that time you caught the first catch, you labeled half the population. Okay? So that's kind of the way the maths works out. All right, let's pinch out and see where we got to. Right, now we have covered levels of biodiversity, how to do Simpsons biodiversity index, and different ways of sampling animals and plants. So before we move on, I think it's worth saying that actually when we're looking at sampling today, there are other more modern methods that we might use. Most of these methods in the textbook and in the specification are about sampling invertebrates, little tiny creepy crawlies, bugs, insects, and arthropods. But actually, if we were looking at sampling larger animals, like tigers in the forest in India, we're not going to use a pitfall trap for a tiger. That's crazy. What you'd probably do is you would set digital cameras that trigger remotely when anything moves past the trail. So camera traps is a really, really big method of sampling used. And you could also do all sorts of crazy stuff, like I've seen DNA sampling. You can actually like take samples of water and work out what animals are swimming in the water, because DNA technology is so sensitive. You could do noise sampling. You could put a microphone in the water and listen to the sounds of whales. And if you hear the whales, they're somewhere around, and then you know they're there. So there are lots of other methods of sampling, which you can research for yourself, but they're not in the specification. Okay, let's move on. Okay, so now let's look at the actual benefits of biodiversity. We've talked a lot about it, but why should we really care about biodiversity? Well, we can first of all kind of divide this up into a few key different areas. So let's look at the economic benefits, the ecological benefits, and the aesthetic benefits to biodiversity. So first of all, lay out our subheadings: economic, ecological, and aesthetic. Let's, I guess, take the easiest one first. There is an aesthetic effect of biodiversity on humans. If you go to a place that is very biodiverse, most people think that they get a feeling of calm, they get a feeling of satisfaction, and it has even been shown to do stuff like lower your heart rate if you're stressed and that sort of stuff. So we can call this the aesthetic effect of biodiversity. So there's an effect on well-being. Was well-being one word or two? I'm going to hyphenate it. Well-being. There's an effect on well-being. It looks nice. It's good to be in. So that's one effect. But even if we didn't worry about that, let's now look at the ecological arguments. So there's a few things here, and generally they center on, number one, interdependence. And the idea of keystone species. So first of all, interdependence. Interdependence is the idea that ecosystems are complex systems, and if you take out parts of the ecosystem, if you take out one part, it might not affect it. If you take out two parts, it might not affect it. But sooner or later, if you keep taking out parts out of the ecosystem, it's going to become unstable. I saw an analogy the other day in a film, I think it was, of taking individual screws out of an airplane while it's flying. You can probably lose a screw or two, and it's not going to cause the airplane to crash. But sooner or later, if you remove all the pieces, it's going to kind of crash down. So interdependence, I'm going to say removing species makes, kind of cramming it in here, system less stable. Okay, and this is exemplified by things like examples of keystone species. So what is a keystone species? A keystone species is, let's define it, it is a species, let me zoom in here because I'm kind of trying to cram this in, aren't I? A species that has a disproportionate effect. Disproportionate effect. And let's look at two examples just to kind of elaborate. Wolves in Yellowstone. And beavers. Okay, so if you haven't seen the Wolves in Yellowstone video, I highly recommend you go look at it. right now if you want. We'll come back to the video or at the end. But if you go into YouTube and you search in how wolves affect rivers, it's a fascinating video. Basically, before the 1990s, all the wolves in Yellowstone were gone. They'd been hunted to extinction. When they reintroduced wolves in the 1990s, it had a dramatic effect on the actual landscape of Yellowstone because it meant that the deer were under threat of predation. The deer didn't graze as much, more trees grew up, more animals came back with the trees, and basically the whole ecosystem was changed. So the wolves are a keystone species, a top predator, and when they were reintroduced, the whole ecological health of the ecosystem got better. Beavers in the UK are being reintroduced. They're being reintroduced in Scotland. They're being reintroduced in Devon. And they are ecosystem engineers, which means that they build their little dams, create wetlands, which increase biodiversity by providing habitats for fish and also for sort of birds that like to kind of hunt in the water, like kingfishers and that kind of stuff. So these are two examples of keystone species. And we keep finding really more and more examples of keystone species in every ecosystem we really look at. So let's make a little bit more notes on this. Let's go over here. I'm just going to write a little side note up here. Oh, I've kind of already written this down. I'll write it again. So more biodiverse equals more stable and more resilient. Plus more resilient. So more resilient to things like climate change, for example. So climate change and biodiversity are very linked. We'll look at that later. But if an ecosystem is under threat from climate change, better that it's biodiverse, more stable. So, okay, if we discount the aesthetic effects, if we even discount the ecological effects, we don't care about the environment, there are concrete economic benefits to maintaining biodiversity. And we can look at kind of two areas. First of all, we can look at something called ecosystem services. And these are the services that ecosystems provide us, humans, or in our society. Ecosystem services. Now, basically, up until quite recently, we took these services for granted. But economists are now starting to try and kind of like value these services. Like if we had to pay for these services, if the ecosystem, if the environment wasn't doing it for us, how much would we have to pay a business to do this? So we can kind of put a value, an economic value on these. And there's a ton of things that the ecosystem does for us. So, for example, there is atmosphere cleaning. There is fresh, clean water. So water purification. There is soil formation. That is a massive one. I bet you haven't thought about soil very much before, but it is a big deal. If there was no soil, you would not have any crops. You couldn't grow anything. And soil takes a long time to form. The ecosystem slowly builds it up over time. And it's very difficult to kind of replenish if it gets washed away. So soil formation. Pollination. Now pollination, sometimes the ecosystem does not provide it. Sometimes we've so removed pollinators from the environment that we have to get in pollinators. So for example, in the Central Valley in California, Google it, they grow like pretty much, I can't remember, it's like over 50% of the world's almonds in this one valley. And all the trees bloom at one time of the year, and there's just not enough bees at all to pollinate all the trees. So they basically bus in huge numbers of hives in order to pollinate those bees. And they have to pay for it. So there's kind of an example of the economic value of this sort of service. Nutrient recycling. So this is things like when fungi and bacteria break down dead organic matter and return it to nitrates, phosphates, that kind of stuff in the soil. Finally, one I almost forgot about, oxygen. Photosynthesis provides us with oxygen, gets rid of CO2. Pretty important. So all these things are called ecosystem services. And I will kind of box this in here. Those are ecosystem services. Now, another thing that's kind of worth just quick noting is that things like beavers down here, they also can provide direct ecosystem services, which are kind of have been overlooked but are kind of becoming valued. Flood prevention. Okay, so beavers actually prevent flooding by slowing the movement of water from kind of high ground down to the low ground, because these dams kind of block the water flow, slow it down a little bit by little bit. Forests also do this. They act as a sponge. So when you have landscapes that are barren, just grassland, water flows off, can cause big swells in the rivers, cause flooding in places like the UK. Whereas if we had forests, beaver dams, that kind of stuff, flooding would be less of an issue. So some councils, some places are looking into reintroducing beavers as a means to prevent flooding. So those are some economic reasons. The other economic reason is basically direct benefit. Okay, direct benefit. I'm going to kind of go down here to fit it in. So there's a direct cash benefit, which we don't have to kind of estimate the kind of economic value. We can actually, just look it up. So chemicals in pharmaceuticals. Come from somewhere. They come from the ecosystem. Often the rainforest. Oops, I can't spell. Pharmaceuticals. Pharmaceuticals. Timber and products. Forest products. Okay, these have a direct cash value. Okay, so those are some of the reasons to conserve biodiversity. But what about things that affect biodiversity? What are the main things that affect biodiversity? Well, let's look at them. There's about sort of 3 major things that affect biodiversity. So I'm going to do my arrows going the other way, just to mix it up. Number 1, what affects biodiversity? Human population affects biodiversity. I'm going to zoom in a little bit more. This is the big one. The human population affects biodiversity. And we're going to draw a little graph here because the human population, over the course of like a lifetime, not my lifetime, but a lifetime, David Attenborough's lifetime, I hope you guys know who David Attenborough is, it has gone from basically 1 billion to 7 billion. So, David Attenborough was like 90 years old, but still, in one lifetime, the population has really just skyrocketed. We are almost on an exponential trajectory. So at the moment, the human population is about 7 billion, and it's predicted to kind of top out probably at 10 or 11 billion, sort of based on expected kind of birth rates. It all depends on like birth rates and how much they kind of, increase, or how much they're not increasing, how much they decrease and how quickly they decrease. Basically, as countries get richer, generally speaking, birth rates drop. For example, in the UK, I'm not sure what the actual birth rate is now, but I think it's maybe just about replacement rate, which is 2.1, I think. So like as many people are born as die. And it kind of all depends on that. So the human population has gone from 2 to 7 billion. Did I say 1 to 7? I did, didn't I? So it's gone from 2 to 7 billion in one lifetime. So 90 years. And it's predicted to keep going. So why is this such a big deal? Well, people need buildings to live in. So the fact that there's more people means there's more buildings. They need infrastructure. They need roads, power stations, all that kind of stuff. Infrastructure, bridges. They pollute. They deforest. So pollution includes things like carbon dioxide. They require energy. Okay, I think this is actually a big one, which I forgot to talk about last time I did this mind map. This is the second time I've done this mind map. Energy demands. People's number of gigawatts or whatever they consume in a year, or megawatts, it is increasing because we have more stuff, we have more technology, we consume more electricity. So energy use is growing as well. So human population drives the loss of biodiversity and is linked to stuff like agriculture, because with more population, we need more food. So agriculture is another big one. I'm going to put this here. So agriculture can be divided into crops, animals, I'm going to write crops plus feed. Okay, so land that we grow crops on, like crops for human consumption, like corn, and also land that we grow crops for animal consumption. So we might grow corn to feed chickens, or we might grow soy to feed cattle. And actually, this amount of land is huge. We use far more land to grow crops for animals to eat than we actually use to grow crops for human consumption. So with increasing agriculture, increasing land used for agriculture, we reduce biodiversity because most agriculture is a monoculture. A cornfield or a soy field would just basically have soy plants in it. And the rainforest that was once there, a lot of soy is grown in Brazil, is gone, so the biodiversity is way reduced. And also agriculture uses chemicals which can reduce biodiversity. So we're talking about, I'm just going to kind of, doesn't look super neat, but I'm going to branch over here. So we're talking about fertilizers, can produce eutrophication, which is not really on the syllabus, but you might have heard it if you do geography. Eutrophication, we've got fertilizers, we've got pesticides and herbicides, and things like that. And these have consequences, and they reduce biodiversity. So we've got human population, we've got agriculture, we have climate change. Climate change is a massive driver of biodiversity loss. Where am I going to put this? Here. Now, climate change is, of course, linked to increasing human desire for energy, basically CO2 and methane are the main greenhouse gases, and their concentrations are ever increasing, and it leads to increasing global temperatures. So the world is becoming hotter, plus maybe drier in some areas. Australia, California, huge droughts at the moment, have been going through multi-year droughts, the longest that we've ever seen. This is leading to forest fires. So I'm going to put that over here. Hotter, drier, fires. This is changing the landscape. Some people think that if it gets any hotter and drier in Brazil, basically the entire Amazon rainforest will burn, because we'll just have sort of positive feedback that will kind of just run away. We've got changing weather patterns. It may be that the rains come at different times. And with climate change, we may have, sometimes animals might move further north if it's getting hotter. It might move to more northerly or more southerly latitudes, basically close to the poles, as they kind of move away from the hot temperatures. But sometimes there are barriers to migration, which means that animals can't do that. So I'm going to again put this over here. I'm just going to cross over here, barriers to migration. A really good example of this, I'll just draw a little quick picture here, is like sometimes you get these like species that live on a mountain, right? There's a mountain, there's a sort of volcano, possibly in the Virunga National Park in Rwanda. And mountain gorillas, live, a certain area. Then maybe they live down here in this kind of green area. If it gets hotter and hotter, well, they can just move up, can't they? They can move up. But what happens if it gets even hotter? Can they move up any higher? No, they can't. They become stuck on the mountain peak. And some scientists have called this an escalator to extinction, because eventually there's nowhere more for them to go. and they're stuck on the top of the mountain, and it becomes too hot for them there, and then they're extinct, they're gone, escalated to extinction. So really, all of this, sadly, leads to extinction. You know, that's what we're really talking about. We're talking about biodiversity loss. We're talking about animals going extinct. So there's hundreds and hundreds and hundreds of animals going extinct. It's really, really sad. You should watch the documentary Extinction: The Facts, David Attenborough. It's on iPlayer. But you know, things like the dodo, what happened? Humans ate it. Things like the polar bear, not extinct yet, but is it going to be extinct because of climate change, decreasing ice, sea ice? So there's countless examples because of climate change, and we can also things like introduced species. So sometimes humans will either deliberately or accidentally introduce a species to a new area, and it causes chaos. Like in Hawaii, there's a lot of introduced vegetation that is outcompeting the native vegetation. So I think there's a lot of plant species on Hawaii are in danger of extinction. Okay, right. So that's pretty grim. That's horrible stuff. How can we solve it? Can we protect biodiversity? Yes, we can. Conservation. There are many different ways and strategies of conservation, and that's where we're going to go next. So scroll across here, and we're going to talk. I'm going to pinch out a bit so I can sort of see how much space we've got left. Yeah, we can do this. So we're going to talk about three major ways of conserving biodiversity. We're going to talk about in situ, ex situ, and we're going to talk about laws. And we're going to have to try and take up this whole little box up here. So I'm going to draw a big line up to there. Let's make it thicker. Okay, so in situ. And then we're going to talk about ex situ. And we're going to talk about laws also. You can fit that down in here. All right, let's talk about ex situ first. Okay. So ex situ conservation means conserving animals not where they normally would be, in some form of human-built place, shelter, something like that. Okay, so basically it involves kind of like confinement. And a kind of monitoring. So what kind of things, I'm going to zoom right in. What kind of things? Well, we can have things like, what I'm going to do, zoos. We can have things like botanic gardens, kind of like a plant zoo, if you like. Botanic gardens is like a garden where you've got a wide range of species planted under controlled conditions, maybe some greenhouses, that kind of stuff. Kew Gardens is a big one in London you could visit. And seed banks. Seed banks is a little bit like a ultra compact botanic gardens that houses only the seeds of the plant, and there are big places like this where the seeds of lots of organisms are stored. Here's a picture of one. It's called the Svalbard, I think, like Global Seed Bank or something. It's built in a place supposedly, even in a nuclear war, all the seeds stored there under kind of frozen conditions would survive. And if there was like a nuclear war, we could even use all those seeds to like replant the world, which really hopefully we won't have to do, but that's the idea behind it. Okay, so ex situ, those are some examples, but why would we do this? What are the advantages and disadvantages of this? So let's go across here. I'm going to kind of go to, let's do the advantages. Advantages of this strategy. Well, we can protect the things that are there. If we've got the northern white rhinoceros, which is pretty much gone, to be honest. There's only 2 left, and they're both female. But if we wanted to protect the northern white rhinoceros, maybe it's best to have them in a zoo where poachers cannot get to them. We can monitor them. We can breed them under controlled conditions. We can sort of have a breeding program where we can sort of carefully, I don't know, maybe choose which organism to breed, which organism to kind of can maintain genetic biodiversity and prevent inbreeding. We can do research. We can research the animals that are being conserved in situ, and potentially if there are still animals out in the wild, then that research can be used to sort of protect those or to help those as well. And finally, we can use these animals for kind of education. So this is kind of, education, not of the researchers, but public education. So like maybe you've been to a zoo in your life, maybe you enjoyed it, maybe that gave you some sort of appreciation for wildlife when you were, a young child. I'm not sure, but that's the argument. Okay, so there are some minuses to this and some negatives to this. I'm just going to do that in red. It's kind of how my brain works. Green for positives and red for negatives. So what about the negatives? Well, it's hard to keep enough of the species to maintain biodiversity, so it tends to result in low biodiversity. And I'm talking really about gene pools here, so I'll put low biodiversity brackets genetic. Because remember, genetic biodiversity is the percentage of polymorphic gene loci. And that can tend to decrease if we're reliant on ex-situ conservation. So often animals in ex-situ conservation kind of environments demonstrate abnormal behavior because they're not in their normal environment, and they, in poorly designed zoos, for example, animals display repetitive behaviors that's pretty much a sign of like psychological damage for the animals. So modern zoos, good zoos, do a lot of things to try and enrich the animals' environment, providing them with like new toys and new stimulation on a daily basis. But like in old style zoos, like concrete cages, the animals kind of went a bit crazy. It's sort of horrible to see. So abnormal behavior, but this might be also kind of less dramatic abnormal behavior, but like abnormal breeding behavior. So it might be very difficult to breed an organism in captivity because you haven't quite got the right mood for the breeding to occur, for example. And ultimately, it's temporary. What I mean is, you can't conserve animals in zoos forever, right? And for the next 2,000 years. It's just not a sustainable long-term solution. So most people say that having zoos and ex-situ conservation basically just needs to conserve animals for the next, 100, 200, 300 years until we basically get our stuff together and fix the planet, basically. So that we'll just make a note of that now. So ex-situ plus in-situ. In-situ is in the wild. And if we can kind of use these two, ex-situ and in-situ together, there's a possible repopulation angle. So we can potentially repopulate, ecosystems that have been destroyed, and then someone comes along and tries to rebuild them, replants the forests, allows plant biodiversity to reestablish, and then they might reintroduce, let's say, tigers, rhinoceroses and stuff like this. I'm thinking about some nature reserves in northern India that I've read about. And this is called rewilding, and this is a really big thing in conservation at the moment. It's kind of something to get excited about. There is a big rewilding movement in the United Kingdom. I can't remember the name of it. There's one estate where they're trying to rewild it. I think it's in East Anglia somewhere. And they're basically just like not farming it, just letting it kind of go wild. But they're also reintroducing big grazing animals to like turn over the soil to reestablish biodiversity. So this has a lot, there's a lot to do with Keystone Species here. And it's a big deal. Get excited about rewilding. If you want to, go research it. So that's in situ and ex situ kind of together. So, but what are further examples of in situ conservation? Well, often there's some form of wildlife reserve. So when we're building a wildlife reserve, there's a few things that you need to kind of like think about, questions when you're building your wildlife reserve or establishing its borders. Is it comprehensive? Like, does it have all the different types of, different sort of subtypes of habitat that you might want in your reserve? Or is it just one type of habitat, or does it contain many kind of linked habitats together? Is it adequate? Is it big enough? Okay, adequate in size. Like, it would be silly. I can't spell that. I need to rub that out. It would be silly to build a reserve that, did not have sufficient area to support, one tiger. Right? You can't build a reserve for, like tigers and have it like, one city block by one city block, given the territory that a tiger needs. Okay, so is it adequate, bracket size? Is it representative? Of the kind of place you're trying to conserve? Again, this is kind of to do with comprehensive. It's sort of similar. Comprehensive, adequate, representative. And I suppose another thing which we're thinking about more and more is maybe sort of migration corridors. So this is, or just animal movement corridors. So this is the idea that if you have, like, let's say in an island like Madagascar, you might have like 7 or 8 major reserves. But if animals can't move from one to the other, then it puts them at risk of what's called genetic erosion. So the biodiversity gets less and less, the genetic biodiversity. So there's more of a movement now when you're establishing wildlife reserves to have small channels between the reserves where animals can kind of come and go into the reserve and out of the reserve, bringing in new genetic biodiversity, potentially new species, and so on. And that again makes the whole biodiversity of that country or that continent more stronger and more resilient. Okay, so that's wildlife reserves. And in the UK, there are a couple different types of wildlife reserve that you should know about. Because these are on syllabus and they come up. So in the UK, we have a couple different things. We've got national parks. Gosh, I should know more of these. Like, I think the Lake District is a national park. We've got national nature reserves. We've got sites of special scientific interest. And these are quite small, these ones. Like, there are thousands of them. So this could be like anywhere where scientists and ecologists have identified that, like, some rare bird, insect, whatever is breeding, then this could be designated as SSSI. Or places where like a wild orchid has been spotted that is quite rare, this could be designated an SSSI. So they can be quite small. And then we also have things called marine conservation zones. Right, okay, so one extra thing to think about when we're thinking about wildlife reserves and sort of in-situ conservation is it preservation or is it conservation? And pretty much preservation is if there is minimal or zero human interference. So preservation is literally like fence an area, potentially patrol the outside of the area to make sure no one goes in, and then just leave that area to it. No humans go in, no trails, no tourists, no management of the ecosystem, nothing, just let the world get on with it. Whereas conservation is more active management, so you might have tourists coming in, you might have regular tours, you might have scientists researching, you might even fell some trees and maintain the landscape and plant some trees and kind of manage it. Okay, so preservation is, we can just sum it up as preservation, no humans, none. Okay, and conservation, human influence. Okay, so maybe some of these national parks, national nature reserves, sites of special scientific interest, and marine conservation zones might kind of sit a different, well, depending on the characteristics of the specific ones, they might sit more towards preservation or more towards conservation. There's one further bit to do, isn't there? Laws about conservation, and these come up in questions, and people basically always forget them because, well, I didn't train as a lawyer, so I don't find the laws around conservation so interesting. But I'm trying to because they're important and they save, say, biodiversity. So, what laws do you need to know about? Well, there are some international laws, 2 of them, and there's 1 UK law. Okay, so first of all, the international laws. We've got CITES. And I don't know what this stands for, but it's the something International Trade in Endangered Species law. So CITES regulates trade in endangered species. Or forbids. So for example, ivory. Okay, ivory you cannot buy because it is produced by poaching elephants. So internationally, you are not allowed to buy ivory. You are not allowed to buy rhino horn, although it should be stated that clearly you can buy it. Okay, it is still illegally sold, often for uses in medicine. It has no medicinal value whatsoever. It is equivalent to taking fingernail shavings for cancer, and it's kind of sold by people who are trying to get money, from people who are in desperate circumstances. So the CITES agreement prevents trade in endangered species, things like pangolin scales, ivory, all that kind of stuff. There's the Rio Convention on Biodiversity. Rio Convention on Biodiversity. And this, I think, was a big biodiversity conference. I forget the exact year, but it was in the 90s, like 1994 or something. And basically all the governments got together and were like, biodiversity is a big deal and we should try to protect it. So it's more of an agreement, an international agreement between governments saying, we will try to do better. We will try to follow ideals of sustainable development. But I don't really think that any of those laws were sort of binding. And it was more just like a friendly agreement between governments to try and do better. But I don't think they really have done any better. And then there in the UK, there's the Countryside Stewardship Scheme. So that's right, UK here. Countryside. I can't say this word for some reason. Countryside Stewardship Scheme. So this is. Farmers can sign up to this scheme and they can get grants and funding to basically farm more sustainably. For example, you might have a farm, you've got a field here, you've got a field here. In the middle, you could either have a fence or you could have a hedgerow. Now, a fence is cheap. We just put poles in the ground and wire up, but nothing lives on a fence. Whereas if you put a hedgerow, it requires a bit more management. It might cost a bit more, but it's a habitat for small insects, small mammals, birds, etc. And it may provide a corridor between patches of woodland that are separated by a few hundred meters. So farmers can sign up for the scheme, and it promotes more ecological practices. So those are some of the laws around conservation. Right. Okay. Let's zoom out and review. Okay. So to start off, we talked about the 3 different levels of biodiversity that you need to know about: genetic, percentage of polymorphic gene loci, habitat, how many different habitats there are, and species, biodiversity, which can be subdivided into species richness and evenness. A good way of measuring this is Simpson's index of biodiversity, which takes into account both richness and evenness, and there's a bit of maths involved there. In order to calculate biodiversity, though, you need to do some sampling. Now, with plants, often this involves quadrats, which can be used to count plants or to measure percentage cover. You can do sampling randomly, using a random number generator, or non-randomly. And non-random sampling is more often done when you know something about an area, you know there's like different parts to an area. You could do opportunistic, stratified, or systematic sampling, such as taking a transect. With animals, typically we're talking about sampling invertebrates. You can use pitfall traps or sweep nets, Tulgren funnels to extract insects from leaf litter, or you might do light traps. That's something which I think I missed. Light traps where moths fly to a light bulb and then you can kind of collect them. We can also do catch and release estimates of population size using some of those methods. So why should we be caring about biodiversity? The benefits of biodiversity can be subdivided into economic, ecological, and aesthetic reasons. Basically, if we didn't have biodiversity, ecosystems would collapse, and a lot of the things that we take for granted would not be provided to us. But biodiversity is under threat. Human population, climate change, and the demands of agriculture are all affecting biodiversity, and ultimately this leads to extinction of more and more species. In order to protect biodiversity, we can do conservation, or we can also actually do preservation, letting nature be, if we can kind of protect people from or stop people going into an area. But if we're conserving, we can do ex-situ conservation in things like zoos, botanic gardens, and seed banks. There are pluses and minuses to that. Or we can do in-situ conservation, which is basically letting animals live where they should be living. And there are kind of different kind of ways of doing that, ranging from total preservation to more actively managing and conserving those areas. Finally, we can use legal systems to conserve animals. CITES regulates trade. The Rio Convention on Biodiversity promotes sustainable development, and laws in the UK, such as the Countryside Stewardship Scheme, aim to promote more sustainable practices in agriculture. So that is all of the OCR A-Level Biology module 4.2 on biodiversity. I hope you enjoyed it, and I'll see you next time.